Hey, Bio 30s, it's Manchak here, and we are ready to get started on our next unit, which is Unit C, and it's a doozy. It's taking us all the way through a big discussion on cell division, and then we get into something called classical genetics, where we're going to talk about Punnett squares and heredity and all sorts of stuff like that. And then the last bit of the unit is going to cover DNA and how the structure of DNA, protein synthesis, and all of that kind of stuff. So we've got a big chunk of a unit to go through here. Normally, what I do, just so you guys are aware, is I split up this unit into 3 parts. And if we were going to be doing big exams, then I would divide it into 3 exams so that you guys would have a little bit more manageable pieces to bite off here. But since we're basically going concept by concept, it shouldn't be too bad for us to digest this. So what I've got up on the screen right now is just a quick breakdown. This shows you the concepts that we're going through over the course of the unit. This video, so there's 11 concepts in the unit. This video is going to cover concept C1, which is going to take us through mitosis and the cell cycle, and talking a little bit about the importance of understanding mitosis. And then our next video will move us into concept C2 and C3. Probably I'll combine those together. And then we are going to get into doing the remainder of the unit after that. So we'll get into things like inheritance, dominance versus recessive. We'll do Punnett squares. We'll talk about other patterns of inheritance that we may not look at, like sex-linked inheritance, those kinds of things, crossing over, gene linkage. And then once we get to concept C9, C8 is sort of a transition and simple concept into transitioning into DNA and sort of where we are now and what we understand about DNA and its structure. So we've got some stuff to get through here, guys, and it's going to take us a little while to work our way through it. Oops, where am I headed here? I want to be here. Where we're headed now, and I realized I made a mistake here, so I'm going to just change that to C1. For our C1, basically we're going to take a little bit of a work through starting out talking about chromosomes, and that's going to become the basis of what we talk about through pretty much all of this unit, and even a little bit of chromosomes in the last unit, Unit D, that we're going to get into later on. We'll be doing Unit C, by the way, all the way through to about the 3rd week of May, I think, based on our schedule that I posted on the Google Classroom. But we may go a little faster. It depends on if I can squeeze some concepts together. And I'm going to try to start to shorten things up a little bit for you too, because I know listening to me talk for 40 minutes at a time can be a little bit boring when there's no face-to-face interaction. So some things that I want you guys to really pick up on from this lesson. Pay attention to chromosomes and the language I use to describe chromosomes and what they are, and try to wrap your head around what a chromosome is, what it's made up of, and that organization between DNA, genes, chromosomes, genome, right? We're gonna kind of move our way through that progression. It's important to understand the cell cycle and what the cell cycle is all about, and so that'll be one of the first things we discuss after chromosomes. Then please pay attention to mitosis. Understand what the stages are of mitosis, and please pay attention to what's happening with the chromosomes, because when we move on to concept C2 and we're dealing with meiosis, I really want you guys to be able to see the differences and similarities between the two. And then we'll do a little bit of discussion in this lesson on the applications of meiosis and mitosis, pardon me. And really, honestly, I don't need you guys to know a ton about this stuff. I put a lot of it in more for interest's sake, stuff that I think is really cool, like stem cells, things that we should really be paying attention to. I'm going to have a whole whack of resources here for you guys for this section. Again, you do not have to look at all of these. If you feel like this video lesson was enough, by all means, that's enough for you. But I would recommend giving a few of them a view, particularly this Amoeba Sisters video. They do great jobs with animations, which is more than I can do on an explainer here. And then there's a bunch of other stuff. Some of these things are more pure interest sake, like the stem cell stuff at the bottom here, and the Veritasium and Minute Earth videos. I think those are great. They're worthwhile watching, but they're not directly on topic to what we're talking about, but they do link strongly to it. Then you guys will have some practice questions out of the textbook, and then I'm going to set you guys sort of an internet assignment where I've put together, and I usually do this anyway, some websites to visit that do a good job of showing you what's going on in mitosis. And so I'll do that as an assignment, and it's going to replace the worksheet that I would normally offer in this section. You'll do a practice quiz and then a summative quiz, and then that is concept C1 done and done. So what we're going to do now is I'm going to jump into the lesson for you guys. We'll run through it as quickly as we can. And then as always, if you've got questions when you're done, go ahead and ask. If you haven't downloaded them yet, I have put the notes for concepts C1 through C3 on the Google Classroom under Unit C, which is look for resources, notes, and worksheets, and you'll find those notes in there. Print them off or have them open, highlight, underline, make notes on spots where you think you might need to ask questions, those kinds of things, and I can answer those questions for you. So we're going to jump into this unit here. And really we're going to be covering part 1 of this unit is mitosis and meiosis and trying to get a good understanding of that. So we'll look at the two types of cell division, which are mitosis and meiosis, and talk about chromosomal behavior during that division. And then the other things that we're getting into, like I said, is classical genetics and trying to understand what that is, and then the basic structure of deoxyribonucleic acid, or DNA, protein synthesis, mutation, those kinds of things. So that's right at the end, D9 through D11, I believe. So for this particular part of the unit, our main objective is to describe the processes of mitosis and meiosis, with the focusing questions: what cellular processes allow for growth and reproduction of an organism, and that's our cell division plus for part of the cell cycle, and then what regulates the transmission of genetic information from one generation to the next. That's kind of our lead in to the rest of this unit from C4 on. If you want to take a few minutes, you can have a look at your textbooks, page 554 and 555, questions 1 through 5. Those are some good introductory questions to have a look at to get you thinking about this cell biology stuff again that you may not have thought about much since Bio 20 or even Science 10, but I'll leave that up to you. So what we're going to talk about first here is the cell cycle. And the cell cycle is the sequence of events that a cell goes through, if you want to think of it in its life cycle. And so we're going to talk about the ideas of interphase, mitosis, and cytokinesis, and we'll talk about what those three things are and how we relate them. And I've got some great diagrams that are in your notes and in the presentation here that we can work our way through. So I usually take a few minutes to ponder some of this. This is based off of what's going on that Are You Ready stuff we just talked about. You know, things like what are the advantages of being multicellular? What are the advantages of reproducing asexually and sexually? If 22 chromosomes are found in the muscle cell of a mouse, how many chromosomes would be in a brain cell, a sperm cell, a fertilized egg cell? We're going to get to all of that stuff as we move through this section, so I'm not going to dwell too much on this. This would normally have been a class discussion, which is a little tricky to do at this point. So I want to talk about the cell cycle, and I want you to consider the cell cycle sort of as a life cycle that an organism goes through, okay, from start to finish, or a cell goes through from start to finish. And each cell goes through this pathway. And you can meet a cell at any point along that cycle, and you can expect that it's going to follow that cycle all the way through, with few exceptions for those kinds of cells. The cell cycle is simply a sequence of events that allows organisms to grow in size, maintain their cells. For example, replace matter or replace those cells after they've been damaged or died, right? If you cut your hand, slice it open, when you slice your hand open, there are cells that are damaged there. Well, the cell cycle, those cells nearby are part of the cell cycle, but they may end up needing to divide and fix that gap up, replace the cells that were lost in order to heal the wound, and that's what's going on. And it's important to understand that this is a continuous cycle. This is something that is going on for all cells continuously, and if the cell cycle stops for all of our cells, we stop as well. Now, before we get into the actual cell cycle, I just want to talk a little bit about chromosomes, because we are going to talk a lot about chromosomes in Unit C. It's sort of the big idea of understanding what a chromosome is, why they're so important, what kind of information they contain, and how all of this links to making us who we are. So, I think you guys know this already, but all of our genetic information is found in our DNA. And our cells each contain nuclei, and in those nuclei is our DNA locked away, providing that blueprint for life that we have. The DNA inside our nucleus tends to be stored in an uncondensed state. What that means is it's not all wrapped up tightly, twisted together, and wrapped around in order to make what we would consider to be a chromosome that we would see later on in this section. Instead, the DNA is in an uncondensed form, not coiled up and twisted, and sort of spread out, making it a little bit easier for the cell to work with that DNA as it goes through reading it for protein synthesis or something along those lines. Specifically, though, the coiled DNA that we find in separate strands in our nuclei are called chromosomes. And a chromosome is very simply a long segment of DNA. Contained in that long segment of DNA are genes. Genes are smaller segments of chromosomes of DNA that code for a specific protein. And those genes themselves are made up of DNA. So DNA makes up genes, genes makes up chromosomes, chromosomes make up the human genome, if we're going from small to very large. Genome describes all of the chromosomes in the human system, or the human individual, and all of the different variations contained within those chromosomes for people here on Earth. So a chromosome is a length of condensed DNA and proteins coiled up to reduce its size. If I wanted to talk about chromosomes that are sitting in the nucleus in the time between cell divisions, we would actually call them chromatin, the uncondensed, not coiled and twisted version of this. But I tend to just say chromosomes. And I want you to understand that chromosomes, we're really talking about segments of DNA, long segments of DNA with certain genes on them made up of DNA. Each chromosome, we have 23 pairs of chromosomes in our regular body cells, and those 23 pairs, why I say 23 pairs is that we have 2 identical chromosomes for each pair, almost identical. The thing that's different is that one came from your mother and one came from your father, and they contain the same genes, those 2 identical chromosomes, but they may contain slight variations on those genes, what we call alleles, which we'll get onto when we get into concept C4. Chromosomes or chromatin are found in the nucleus in eukaryotic cells, and the chromatin condenses at some points in the cell cycle into chromosomes, which actually make them visible under a microscope. And we're going to see some examples of that later on. This is what a stylized diagram of chromosomes might look like. And this would be a condensed chromosome, not chromatin. And there's actually 2 chromosomes on this diagram. Over here would be 1 chromosome, and over here would be the other. And those 2 chromosomes are being joined together by something called a centromere in the middle. This could either be a pair of chromosomes, one from your mother, one from your father, or this could be a pair of chromosomes that have been duplicated so that this side here is exactly the same as this side here. We'll try to explain that stuff a bit later on when we get a little further on into the concept here. A chromatid is represented by this structure here. Okay, what we represented as 1, and 2 chromatids here are being held together by a centromere. We would call these sister chromatids if they have been duplicated, okay, and they're identical to each other. So this chromosome stuff is a little bit tricky, and I'm using words like chromatids, sister chromatids, chromosomes, all of those kinds of things. I'm hoping as we play around with this unit over the next little while, you guys will start to get an idea of what I'm talking about when I say a chromatid, which is actually a chromosome, but it's usually paired up with a sister chromatid, which means they've been duplicated and are identical to each other. We're also going to talk about tetrads and other things, so it's going to get a little bit fuzzy, I think, before it gets clear. And it's okay if it's a little bit fuzzy. Let it be fuzzy for a bit, and we'll fill in those blanks later on. All right, dog's barking upstairs. I think my wife and daughter just got home, so if it gets bad, I'll pause for a minute. The cell cycle, then, is what I want to talk about now that we've talked about chromosomes. And the cell cycle, remember, is that process, that cyclical, continuous cycle that a cell goes through over the course of its life. And it can be divided into 2 large phases that I really want you guys to pay attention to. First phase is called interphase, and then the second phase is called the division phase, or just division. Interphase is where the cell spends most of its time. Now, what's going to be weird here is we're going to spend most of our time talking about mitosis, but don't take that as that's where the cell spends most of its time. It spends very little of its time dividing and most of its time in interphase. Interphase involves the growth of the cell and the preparation for cell division, and that includes in its preparation duplicating the DNA to make sure that when a cell divides, you have exactly the right number of chromosomes in the 2 cells that are created from 1 cell. There are 3 phases, subphases in interphase, G1, S, and G2. And G1 and G2, the G actually stands for gap, but sometimes in my head I like to think of it as growth because it helps me remember what's going on. And so G1 is a rapid growth phase, and it happens right after a cell is divided. Remember, a cell goes from being 1 large cell into 2 smaller cells that are identical, and then they need to grow to get back to whatever they were doing before. So that rapid growth phase helps with that. There's an S phase, S for synthesis, and that's where we get the synthesis of DNA for duplication. So we end up duplicating the DNA at that point. And then the G2 or gap 2 phase, where we see a little more growth and we see preparation for cell division. In general, what I want you guys to understand is interphase is basically where the cell spends most of its time, and it's doing the things that the cell should be doing at that time. So a skin cell in interphase is being a skin cell. So what that means is it's protecting the rest of our body. A nerve cell in interphase is being a nerve cell. It's conducting electrochemical impulses up or down its axon from cell body end to dendrite end. Right? We're doing, those cells are doing what they're supposed to be doing at that time in interphase. They are doing some preparation for division, but that's a small part of what they're doing. So the second part of interphase that I really want you to pay attention to is the division phase. And it involves basically the process of creating two genetically identical cells. And we're going to call it the M phase in the diagram. And I'm going to lump together mitosis, which is that actual, going through and actually starting the division, and cytokinesis. And cytokinesis is that point when the one cell sort of squeezes down, and we'll see this, and pinches off and turns into two cells. Okay, so I'm gonna lump that together as the division phase. So this diagram can be a little bit confusing, and there's sort of two rings on this diagram that we're going to pay attention to. There's an outside ring, and there's an inside ring. Okay, the outside ring is just our two phases: I for interphase, M for mitosis, and cytokinesis. And you can tell cytokinesis happens right at the very end of mitosis. And then these two cells each will enter into interphase, spend most of their time in interphase, and then come up to mitosis, that division phase, which we'll talk about shortly. The inner ring we've subdivided it even more. So we still got our mitosis and cytokinesis right at the end of that. But we're going to spend time in G1, S, and G2. And then I've put on G0 here as well, and we'll talk about what that means in a second. So G1, or gap 1, is where the cells increase in size and they prepare themselves for DNA synthesis. S is where the synthesis is occurring, so we're replicating or duplicating the DNA in preparation for mitosis. Gap 2, the cell continues to grow. It creates more organelles like mitochondria and other things like that to make sure that it's ready for that division when it's done, so that each cell ends up with enough stuff in it. And then it moves into mitosis, which is when cell growth stops and we get an orderly division that leads to two daughter cells. Okay. A note about G0, not usually talked about, but I like to mention it because it's important. G0 is a phase that the cell can sort of pull itself out of this whole cycle, and it's kind of a phase where it rests, and the cell has left the cycle and it's stopped dividing. And a lot of our cells actually do that. Like nerve cells pull themselves out of that and just be nerve cells for a while and don't replace themselves, because if our nerve cells were always replacing themselves, then we would have issues with conducting electrochemical impulses. If that makes sense. So what I want you guys to be able to do from cell phase is or cell cycle, is to be able to break it apart into those two broad categories, and then when we're done this section, break interphase into its categories and break mitosis into its categories, and tell me what's going on in each of those smaller categories or subcategories. So this diagram is just quickly sort of running us through in this direction, this shape of a 7 here, what's going on in interphase. So we've got our first gap phase where we're getting protein synthesis, organelles are being produced, we're increasing the volume of the cytoplasm, right? Look at the size of the cell here compared to the size of the cell here. When we move into synthesis, what's going on is the DNA inside the cell nucleus duplicates. And then when we move into gap 2, or the second gap phase, more organelles are being produced. Notice how there's more mitochondria now than there were here or here. Notice how the endoplasmic reticulum is increasing. We've got now 2 centrioles instead of the one we started with. All of these kinds of things are happening, and the volume of the cell continues to increase. That increase in the volume of the cytoplasm means that we're pumping more nutrients and stuff into it, getting ourselves ready to do that actual division thing. The vast majority of the cell cycle is spent in interphase, and this is the cell activity between divisions, and it's characterized by the G1 and G2 phases: elimination of waste, building proteins, getting ready for mitosis, duplicating DNA, all of that stuff. And by late interphase, the chromosomes have already been duplicated, and it's ready to move into mitosis. That's where we're going to move into now, is we're going to move into mitosis and talk about what cell division actually looks like, and what are the processes that we're going to go into. Remember, I just talked briefly about the cell cycle and interphase. When we get into we're still in the cell cycle. I just briefly talked about interphase. We're going to spend more time talking about mitosis, the division phase, because it's a little more complex and we want to really pay attention to what's going on. As you work through this section, please pay really close attention to what's happening with the chromosomes and how we're making sure that if we start with one cell and we end up with two identical cells, how do we get from one cell to two identical cells that are identical genetically, meaning that they have the same chromosomes in them, in the same arrangement, all of those kinds of things. So the division of a cell and nucleus to form two identical copies of the chromosome and thus two identical diploid cells is called mitosis. That's a big sentence loaded with stuff. So the whole goal of mitosis is to take one cell and turn it into two. Okay? And they're genetically identical. And they need to be diploid. Diploid means that we have two sets of chromosomes. So the set from your mother and the set from your father. So those pairs are together. We have those two sets of chromosomes. For us as humans, that totals 46 chromosomes in 23 pairs. 2 times 23 is 46. Okay, that makes mitosis. The word diploid is going to be important, and we're going to talk about it a lot as we move through this unit. Diploid just means having a full complement of DNA, meaning that you have all the DNA that you should have. In mitosis, the daughter cells receive the same number of chromosomes as the parent cell. So they should be genetically identical. You should be able to go into each of those daughter cells and find exactly the same chromosomes in each of them, and exactly the same chromosomes as you found in the parent cell, the cell that turned into two. Why we care about mitotic division? Why it happens? Well, we've already talked about it once. Okay. Actually, we've talked about it all the way through concept B4. When we were talking about a single fertilized egg turning into a human being, that whole process was regulated by mitosis. Every single cell division that was taking place in that organism, save for maybe some stuff that was going on in the gametes in females, all of that was mitotic division. One cell turning into two identical cells. Okay, so development and growth uses mitotic division to increase the number of cells in an organism. That's how we grow. There's more cells, right? My daughter has fewer cells than I do, right? Because I'm bigger. As she grows, she gets more cells and she becomes larger. It's not like the cells get larger, she gets more cells. Okay, and then we hit the end of our growth and then we stop adding more cells. Well, maybe, we add more fat and things, but those aren't really cells. We do cell replacement. So we shed off skin frequently, right? And so we need to replace those skin cells on the outside. We shed stuff internally as well. Our digestive tract sheds cells all the time. Those need to be replaced. If we hurt ourselves, we have issues. Thinking outside of humans, some organisms regenerate. Some organisms can regenerate a body part, like a sea star can do that. And they regenerate entire limbs based on mitotic division, taking one cell and copying it, and then copying those two cells and those two cells and those two cells and so on and so on and so on until we end up with a new limb. Asexual reproduction is actually done, actually produces new individuals, new organisms as a result of that. If you're a single-celled organism, the only thing that you can do to make more of you is to divide asexually, to take one of you and turn it into two of you. Okay, that's asexual reproduction, and we'll talk a bit more about that when we move on to the next section here. We can divide mitosis into 4 phases, each with characteristic things taking place. And the 4 phases that I want you guys to know are prophase, metaphase, anaphase, and telophase. I like to remember it as PMAT. Okay. When you guys get into university or college biology, you may end up learning PPMAT, where they would go prophase, prometaphase, metaphase, anaphase, telophase. For now, we're not going there. Just remember PMAT. Okay, and each of these are different phases, and there's something specific going on in each of these phases. So prophase is the first true stage of cell division. And what happens during prophase is the following. And I want you guys to know these little bits. First of all, the chromosomes that were uncondensed as chromatin, those chromosomes now become chromosomes. They condense themselves up, they twist, and they tightly pack themselves together and shorten and thicken into chromosomes that we can see inside the nucleus of a cell through a microscope, which is pretty cool. You can't really see them using any standard microscopes when they're uncondensed in interphase. The other things that we can spot with good microscopes is that centrioles appear at each end of the cell. And if you remember, all I've told you so far is that centrioles are responsible for cell division. Well, we're going to see what they do. Those centrioles end up having spindle fibers attaching to them, and those spindle fibers basically help to shove the cell apart, and it starts to take the cell to force the cell to elongate and then eventually pinch off and separate into two separate cells. They're basically acting as guide wires for moving chromosomes, and we're going to see what that looks like in a second. The sister chromatids are joined together at a centromere, and so those spindle fibers actually go, and remember the centromere I talked about over in this diagram, the spindle fibers here attach, one on this side, one on this side, so that we can separate this sister chromatid from that sister chromatid. That's what I'm talking about there. So this is what prophase would look like in our mitotic division. So we don't really talk about the nucleolus, so don't really spend any time on that. The chromatin condenses, and you can see this is uncondensed chromatin here. Hey, we can start to see that characteristic X shape of condensed chromatids. And remember, these are pairs of sister chromatids making one chromosome. Okay. The separation of the entrosomes, don't worry about that too much. And then we get the mitotic spindle. That's this thing right here. So we've got our centrioles. Okay, sorry, the entrosomes are the centrioles. They start to separate. What they mean by that is one is going to migrate over here, the other one is going to migrate to an opposite end. Okay, and then we can get those little spindle fibers starting to grow. So when you got something where you've got these kind of condensed chromatin or chromosomes still inside a nucleus that is starting to break up, you're in prophase. Metaphase is pretty easy to identify if you're looking at trying to identify it. Basically, what happens in metaphase is we get chromosomes that start to move towards the equatorial plate. And if you imagine a globe, imagine a cell as a globe, even though they're not usually spherical shaped. You imagine a globe and you sliced it in half, and then you looked at it that way. This part here is the equatorial plate. This flat bit here is the equatorial plate. So again, we took a globe, we sliced it in half across the equator. We're looking at it here. That face is the equatorial plate. All of those chromosomes, those condensed chromosomes, are going to line up on that equatorial plate and have spindle fibers attached to them. And those spindle fibers attach to the centromeres that are holding those two sister chromatids together. The other thing that happens in metaphase is the centrioles are at the poles. So again, if we have our globe, we're going to have a pole up here and a pole down here at the bottom. Okay, and then all of our chromosomes will be lined up in the middle here. So this is what I'm talking about in two dimensions. We've got our centrioles that have migrated to the other side. We've got our chromosomes aligning along the metaphase or equatorial plate. And we've got our spindle fibers coming off of the centriole, attaching to the centromeres right in the middle of each of these. And we'll have one spindle fiber coming to one side and one spindle fiber coming to the other side of each chromosome. The whole goal is that those spindle fibers are going to basically separate those sister chromatids apart. We're going to see that in the next phase. Anaphase, the third phase, is where we actually get the separation of the genetic material. So we've had those sister chromatids that were moving together, held together by those centrioles, and or centromeres, pardon me, and what's going to happen is those spindle fibers where they've attached are going to pull these guys apart and away from each other. So the centromeres divide, and the resulting chromosomes, formerly sister chromatids, move towards the opposite end of the poles. The same number of chromatids will end up at each pole, making sure that we have 46 chromatids in each cell. So this is what this diagram is showing, right? We've got what these guys used to be joined together, but now they've been wrenched apart by each of their spindle fibers, and now they're moving away from each other. What's going to end up happening then, I think you guys can see this, notice that the all of the organelles have divided here and moved apart, and so we're going to start to see a split that happens right down the middle of the cell here. Okay, so these chromatids now are moving across to opposite poles, and there will be 46 chromatids that move in this direction and 46 chromatids that move in this direction. Telophase is the last phase of division, and basically what happens at this point is those chromosomes now, those chromatids, which we'll call chromosomes now, begin to uncondense. Okay, they become less condensed and they turn back into chromosomes, or chromatin. The spindle fibers dissolve. The cell continues to get longer, and I don't know if you guys have seen that so far, but you know, in prophase, the cell's kind of a little bit fatter. Now it gets a little skinnier, even skinnier, those kinds of things. Then the nuclear membranes start to form around the chromosome again, so we get our nuclear membrane forming around all the chromosomes to contain them. And then ultimately we get cytokinesis right at the end of telophase, and that's where, remember, I've been showing you the cell's been kind of getting wider, longer, and then eventually it's going to do this and pop apart. Okay, so telophase, we're continuing to widen and widen and widen. You guys can see the nuclear envelope is starting to reform. Those condensed chromatids are uncondensing into chromatin again. Our spindle fibers are disappearing. The cell continues to elongate. And then we get to the division of the cytoplasm, or cytokinesis, the breaking apart of the cell, the cytoplasm. And we get what was once one parent cell now becomes two genetically identical daughter cells. And so that's what we're seeing here. The daughter cells divide, we get that pinching off here, and eventually these 2 points will meet each other, and we end up with creating two identical daughter cells. So that is a quick rundown of mitosis. The cell cycle itself, once the division is complete, then each of those daughter cells settle into their own interphase, right? So this one begins its cell cycle over here. This one is going to begin it, and it'll go into G1 and S and G2, and each of them will be getting themselves ready for their next division. This diagram here is just a quick breakdown of what's going on in mitosis, and that's this line here. We're going to come back to this again after we've talked about meiosis, which is this column here, because it's a little bit different. Okay, we go from one cell, right? Notice how these are just individual, and then each of these has duplicated. So we have an extra set or an extra chromatid on each of these. That's that cell duplication in synthesis phase. Then they get separated apart, and you'll notice that this cell down here is genetically identical to this cell. This cell down here is genetically identical to this cell. Okay. What you'll notice here, to contrast with meiosis, which is this column, is that we do get duplication, but the way things line up is different. And what we end up with what we started with at the beginning is not what we started with at the beginning is not the same as what we end up with in meiosis. So we're going to try to get to that in concept C2 as we move along. So that's the cell cycle and mitosis. I was just going to bring you guys back to here and say at this point, I've been talking at you for like almost 40 minutes. If you haven't paused yet to take a break, pause and take a break. I just did and went and grabbed some water. I think it would be a good opportunity now to put me on pause and go and watch probably this Amoeba Sisters video as just another run through of everything we just went through. And then you can try this mitosis animation as well. If you're viewing this on like an iPad or on your phone or something like that, I'm not convinced it'll work because it runs off of Flash, and I don't think we have that on our phones. But hey, maybe I'm wrong. So yeah, maybe go for a pause right now and go check those out. I'm going to jump back into the rest of this concept C1. So I hope you went and looked at the Amoeba Sisters video. If you didn't, that's fine. We'll just continue on here. If you did, welcome back. So this is probably a good point for you guys to jump in as well, and maybe have a look at these questions on page 564 of your textbook, and let me know if there's anything that doesn't make sense about those questions. And then you could also jump in and do the mitosis internet lesson, which I placed as an assignment on the Google Classroom, in place of a worksheet for this particular concept, because I think it's going to work better for you, and you'll just submit it more or less kind of as a lab to me. Probably end up being formative anyway, but I'd like to have a look at it and see where you guys are at with that. So go do the mitosis internet lesson if you do anything at this point. In the meantime, I just want to take a couple of minutes to talk about what's going on with the applications of the cell cycle, why we would care to know about this, and how we're going to practically apply it to our lives in some ways, and where we encounter the cell cycle in our technology or in our scientific knowledge. So the first way is kind of fun. We don't tend to do this. We don't do this as humans, but we talk about it sometimes, and that's cloning. And cloning is the process by which an identical offspring is formed from a single cell or tissue taken from a parent organism. We don't do this as humans. We reproduce sexually, which means that there's the union of a sperm and an egg, and those are haploid cells or gametes that come together. But the idea here in cloning is to have the parent and clone containing exactly the same nuclear DNA. They look basically identical, or they should. And this is going to be considered a form of asexual reproduction because it does not lead to a variation of traits or characteristics. Some organisms naturally do this. They clone themselves. So something like a hydra, which is a little tiny microscopic organism, or a strawberry plant, clone naturally. A hydra sends out a little bud. So if you imagine an individual, and then it has a little bud that starts to grow off of it, and it continues to grow and grow and grow until those two things separate. And where we started was this one. This one is genetically identical to it. So we've got that separation that takes place. Producing runners is the same thing. A strawberry plant will send out a little runner above ground that runs off to another section, and then a genetically identical plant will actually grow off of that. I still haven't figured out this camera thing. I'm still doing it wrong. And a genetically identical plant will grow off of that. So this plant over here, with a runner connecting it to this plant over here, they're genetically identical. Natural cloning is what it is. In animals, cloning can also take place naturally too. For example, if we end up with identical twins. Identical twins, or monozygotic twins, means that we get one fertilized egg undergoing mitosis, and then that early on in the zygote will split into two. And so we would have a fertilized egg up here, and then we have a division that takes place, and then we have two individuals that are being developed from that single sperm and egg union, sharing the same placenta. Okay, which is kind of neat. These two individuals then are essentially identical genetically to each other because they come from the same DNA. One sperm and one egg come together to make two individuals. That's got to be cloning in some ways. Just as a note, fraternal or dizygotic twins are a little bit different. What we end up with is when two different eggs are fertilized separately, and then all those individuals are not genetically similar. They're still twins, but they're not genetically similar. And what would happen in this case, I think you're probably wondering, well, I thought females only released one egg or matured one egg per cycle. Well, what would happen is you end up with two eggs being produced in the cycle, and as luck would have it, both eggs are being fertilized. And we end up with fraternal twins there as opposed to identical twins. We've been cloning things a lot. Plants are cloned quite frequently because we want to create individuals that are exactly the same because they've got some wonderful traits. Plants that sexually reproduce in natural settings can also be cloned as well. So single cells from a desirable parent are cultured in conditions to promote mitosis. And then the plant begins the process of cell division, growing larger. These cells specialize and differentiate, and eventually the plant is formed into, a full plant is formed from one cell. And the new plant then is genetically identical to the parent. What this process does is allow the grower to select plants with predictable and desirable traits. So you might be able to take a plant that is drought tolerant or has better flavor and clone it because you want more of those. And then you grow them without having to worry about variation that's created by sexual reproduction. As soon as you bring in, another sperm or another egg and you bring them together through sexual reproduction, you're going to maybe miss out on some traits that you wanted to end up having. So not all plants can be cloned though. Carrots, ferns, tobacco, lettuce, they all respond well to cloning, but grasses and legumes don't respond well for whatever reason. Animal cloning has been done. I remember when I was taking Biology 30 in high school a lot of years ago, we talked a lot about Dolly the sheep and how Dolly was cloned, and they were actually able to take cells from one sheep and actually clone that sheep into an identical organism. This is something that is quite complex on the surface, and I don't want to spend too much time talking about it because it's not really super important you know it, but it's kind of neat nonetheless. What they do to clone, and there's a diagram, a couple of diagrams here in your notes that sort of show what's going on, is basically they take an organism and sorry, I'm going to jump back here. They take an unfertilized egg and they remove the nucleus and create what's called an enucleated cell, a cell that does not have a nucleus in it. Then they take cells from the blastula stage of a developing organism. Remember, the blastula is that cluster of cells that's been moving down the fallopian tube and eventually implants on the endometrium, and you grab it before those cells start to differentiate into the mesoderm, endoderm, and ectoderm. And they remove a nucleus from one of those cells. Why they do that is that nothing's been specialized at that point yet, and they're grabbing a nucleus, which means it's got a full complement of DNA inside of one of those cells. It's a diploid cell. And then they basically take that nucleus from the blastula stage and they plop it into that enucleated cell. That cell that we've taken the nucleus out, they throw a new nucleus into it. And then that new cell with the new nucleus begins to divide via mitosis, and a new blastula forms and the organism develops towards adulthood. That's what these diagrams are showing is we've removed a nucleus here. Now we have an enucleated cell. We've got a blastula stage bundle of cells taking place here. We pull a nucleus out of that. We plop that nucleus inside the enucleated cell. It grows up, and you get a genetically identical frog, compared to what was going on before. This is just showing a little bit more complex process of cloning a mammal using an embryo splitting technique, which is kind of interesting as well. At this point, I'll just point out to you that I did have a website on here that shows that cloning process as well, with some pictures on it. If you want to check it out, you're welcome to check it out. And then I'm just going to move on to really quickly talk to you guys about stem cells, because I think it's really interesting. Again, this is not something super important you guys have to dig into, but stem cells are really neat because they are cells that have not differentiated yet, and we have stem cells in our bone marrow and in other places in our body. And if we can take those stem cells and tell them to do certain things, we can turn them into certain kinds of cells. And so I've got a couple of videos. I'm not going to spend any more time talking about it, but if you're interested in learning more about stem cells, there's a really neat TED Ed talk here on what stem cells are, and then our ability to reprogram them, regular cells into stem cells, which I think is kind of neat. So something to check out. The last bit I want to talk about here is a little bit of a connection between when mitosis will stop, basically, and why we might grow old, and why it is we grow old, and how that's linked to all of this. And I'm going to let these two videos that I've linked in the Google Classroom explain a little bit more to you about this, because it's a little bit complicated, but I'll run through the basics of it. Each one of our chromosomes has a little cap on it. So if you imagine this is our cylindrical chromosome, this end and this end have little caps on them called telomeres. And these telomeres, or telomeres, basically are these little caps. And what happens is each time a cell divides, a little bit of that cap is lost as it goes through. And so those telomeres get a little bit shorter and a little bit shorter and a little bit shorter each time a cell divides. So it's necessary for chromosome ends to shorten during the chromosome replication, so therefore we have telomeres. Telomeres are nonsensical chunks of DNA. They don't code for any proteins. They don't have any other purpose other than to give us the ability to shorten those chromosomes without losing anything important. Telomeres are disposable buffers that block the ends of the chromosomes. They get shortened or consumed during cell division, but the genes at the ends of the chromosomes do not, because the telomeres are shortening. In our somatic cells are our body cells other than our sex cells, stem cells and some white blood cells. In our somatic cells, the telomeres shorten each time the cell goes through division, and they should almost be considered a clock of sorts, ticking down the number of mitotic divisions that each cell has left before those ends are shortened enough that we're going to eat into genes. Okay, and then we start to get into problems when we start to eat into those genes. Now, notice that I said it appears. It appears that an enzyme called telomerase acts to maintain the length of the telomere in the germ, stem, and blood cells mentioned above. Meaning that those cells, those stem cells, those gametes, and the white blood cells, they don't shorten. What's really cool about this, I think, and terrifying in some ways, is that skin cancer, cancer cells seem to have found a way to no longer shorten their telomeres. And we've found that telomerase is present. So basically what happens is this creates an immortal cell. A cancer cell can divide unchecked. It can continue to divide and divide and divide and divide and divide without ever shortening itself enough that it dies. That's what's scary about cancer, is that cancer is a mutated cell that ends up dividing uncontrollably and causing tumors, and those tumors disrupt the organs that those cells are dividing uncontrollably in, and that's what makes us sick. So, telomeres, kind of cool. Linked to cancer, kind of cool. Go and watch this Veritasium video. He does an excellent job of talking a little bit about this stem cell and telomere stuff, and then have a look at the Minute Earth video. It's quite short. but something that you guys should take a look at. Holy smokes, that was a long video lesson. If you guys made it this far, high five to you, because wow, that was a long lesson. I thought it would take 40 minutes, and we're 15 minutes longer than that already. That is Concept C1, ladies and gentlemen. I hope that was okay for you. I hope you took the time to watch some of those videos. Please don't forget to jump in and do the mitosis internet lesson, because that's going to help you with understanding this stuff. Do the questions out of your textbook. Contact me if you have any questions. Then run off and do the practice quiz that I've got up on the Google Classroom. If you have any questions about that, fire me an email, meet me on my office hour or something like that, and we'll discuss it. Make sure that you do the practice quiz and wait for feedback before you jump into the summative quiz. I need to get you that feedback first. Hey, thanks if you made it this far, you did awesome. Thanks for paying attention. Stay healthy, stay away from each other, as much as you can in this environment. And yeah, we'll talk soon.